This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So today we are going to discuss about animal kingdom, that is fourth chapter of 11th standard. See, animal kingdom. What do you mean by animal kingdom? Or as an introduction, what we can say, you know animals. You have heard about all animals like cat, goat, horse, dogs, all these, right? We have an image. Like when we are saying about the animal, we'll be having an idea how it is or what are all the features of an animal. So when we are looking around, we can see in this environment, we can see different types of animals. And they'll be having different structures, different forms and all. So million of species of animals we can see. And we have studied different types of animals. So from there, like each and every animal we can study individually, right? There are so many number of organisms around us or number of animals around us. So we can't select each and every organism and we can't make a note about that, right? So what we will do, we'll make it as some groups, isn't it? So what we are doing usually, so we said million of species of animals are there. So how to understand them? So for that understanding, for learning about them, these organisms or these animals, we need a classification. So this classification, it is not only for just for an identification. Just to identify that organism, not only for that purpose, we are doing this classification. Then what are all the other purposes or what are all extra information we are getting from this classification? That creates or that forms, the classification forms a systematic position for a new species also, or a newly described species that is getting a systematic position. Among these organisms, it is getting placed somewhere, right? See, if you are telling about frog, that is having a particular position among animals, or if you are telling about dogs or tiger or lion, we will get an idea in which category or under which one it is coming, this lion, tiger or dog, whatever may be. So there is a particular systematic position. It is getting created through this classification. So basic thing or basis of this classification, that is for learning the fundamental features of these animals. Like what and all we can say about the cell organization, right? About the cellular level organization. Then what extra information we'll get from this? Then we can learn about the symmetry, then coelom. Then another thing is body segmentation. That and all we can learn from this through this classification. So all these type of studies, first we are learning about the symmetry, cellular level, or body segmentation, then notochord, then coelom. About all these we are studying. So what help we'll get or what information we'll get? We are making a broad classification of that animal kingdom. Complete animal kingdom will be broadly classified due to this classification, right? See, there are so many other characters also. Like for each group or each species or for each phylum or class, they'll be having particular highlighting characters. So this classification, about that classification, we are going to discuss in this chapter. In animal kingdom, what and all classifications are coming or what and all groups are coming. That one we are going to discuss in this chapter. So for a better understanding, first in this animal kingdom, we'll discuss about their classification. Or we need to clearly understand how we are classifying this organism or what and all, based on what and all characters we are classifying this organism. Okay. So main objectives in this chapter, the main objectives are first categorizing these animals from the unicellular animals to multicellular or to the largest one. We need to include in specific categories. Then description of each group or how to describe them, or by seeing an animal we need to identify the special features and to classify them, or how we are making that classification. About that we are going to discuss. Then we need to evaluate the classification system, our classification system, how perfect it is, or what and all we can learn from that classification. Then on what basis we are classifying, that we can learn from this. Then by seeing the body structure, after learning all these groups or the classification system, by seeing an organism or an animal, we can classify by making out their special features, or by writing about their special features, we can classify them, or we can give a proper position for those organisms. Then each phylum, we'll explain what is phylum, all those things. So each phylum, the highlighting features of each phylum we can write. Then while writing in this chapter, the main thing what you need to note down is, while writing about each phylum, main 5 to 6 points you need to write. Main features. As a common feature, what we can say, see, you know about animals, always some kind of exceptional cases will be there. So when we are telling about the features of a phylum or a group of organism, what we need to do, the common characters, common features which is helping us to make out this organism is belonging to this category or this group or this phylum, that kind of features you need to note down. Then we can classify. After learning this chapter, we'll be able to classify chordates and non-chordates. Okay. So first, let us move to this topic. We know that Aristotle, he is known as father of zoology. Okay, Aristotle, he is called as, he is the father of zoology. And he is also known as father of ancient animal classification. Okay, father of zoology, Aristotle, also called as father of ancient animal classification. Ancient animal classification. So don't get confused, father of zoology and father of ancient animal classification. Who is that? Aristotle. Okay. Then Carl Linnaeus in 1735, he gave a detailed information about the binomial system of nomenclature. We have learned all these things, right? About binomial system of nomenclature. Then in Systema Naturae, in that 4,236 known animals were classified and presented in binomial system of nomenclature of animals. Then in what sequence we did this classification? Do you have any idea about what sequence? In what sequence we have done this classification? See, that sequence, smallest unit from smallest unit of classification. Okay, first the smallest unit, smallest one, which is the basic unit of classification, that is the species. Species, that is the basic unit or the smallest unit of classification is species. Then it is going to genus. Species, genus, then family. Different genus belongs to a family. Then many families will form order. Then many orders that will join together to form a class. So basic unit is species, the smallest one. First, the starting is species. Species to genus, genus to family, then order, then class. Next is phylum. Then comes the kingdom. That is the kingdom. So kingdom is the largest unit. So animal kingdom, that is the largest unit, right? We are learning about a kingdom. So we need to go back now. We are saying about animal kingdom. So we need to start from species, genus, family, order, class, phylum, and kingdom. So for human, how we can write? We can write kingdom, Animalia, right? It belongs to kingdom, animal kingdom, then phylum, Chordata, then class, Mammalia, because he belongs to mammals, right? Then order, primates. Then which family? Hominidae, genus, genus and species. That human beings, how we will write? Scientific name Homo sapiens. In that Homo sapiens, if you are writing Homo sapiens, Homo sapiens, this is the genus name and this is the species name. First one is the genus name and second part is the species name. So Homo sapiens, in that it's very clear Homo is the genus name and species name is sapiens. And which family it belongs to? Hominidae. Order is primate, then class Mammalia, phylum Chordata, kingdom Animalia. Okay. Then let's see which are the important phylum. the whole animal kingdom, it is a very large group that belongs to 31 phyla. Animal kingdom, this animal kingdom, it consists of 31 phyla. Animal kingdom that include or that consists of 31 phyla. In that, among these 31 phylum, 10 phyla are major, 21 minor ones are there. Okay, 21 major phyla and 21 minor phylum and 10 major phyla are there in animal kingdom. Okay, so the important ones, just we need to learn everything in detail. So just I'll explain each one later. Right now, first phyla Protozoa, then Porifera, Coelenterata, Clinophora, Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. These are the major phyla. With the example, we need to learn each phyla. Then, next, let us see the basis of classification. On what basis we are doing this classification? See, here you can see different types of animals. See, different types are there. So on what basis we are doing this classification? There are some fundamental features for this classification. So almost all these are based on the cells, body symmetry, nature of coelom, patterns of digestive system, circulatory system, reproductive system. Based on all these, we are making different groups. So these features, mainly we are using these features as the basic characters of animals for their classification. And we are going to discuss those features, main features we are going to discuss now. So first character which we are going to explain is levels of organization. Levels of organization. So what do you mean by levels of organization? See, in animal kingdom, you can see different types of animals. We said it already. Already we told there are different number of animals in animal kingdom. So each group or each member will be showing different pattern, right? Or organization of cells, that cell organization in each group of animal will be different. Like in sponges, the cells are arranged as loose cell aggregate. Here you can see cells, you can see as loose cell aggregate in sponges. That means they are showing cellular level of organization. Okay, so first, let us see there are three types, three levels of organization. One is cellular level of organization. Second type is tissue level of organization. Third is organ level of organization. Okay, first one is cellular level, then tissue level and third one is organ level of organization. So there are three levels of organization. In first one, the cellular level of organization. So you know that animals, they are multicellular. So there are different patterns of the cell organization. See, in sponges, we said already that they are having loosely arranged cell aggregates. Loosely arranged cell aggregates in sponges. Okay. And there is some kind of division of labor, like some group of cells or some cells will be performing for a specific activity or particular activity. That's why division of labor. That labor is divided for particular cells. Okay, so that is called as division of labor. So they are exhibiting cellular level of organization. Up to some extent, it is exhibiting like that. And in coelenterate, the cell arrangement is more complex, little more complex. Compared to sponges, in coelenterate, the cell arrangement is little more complex. So there the cells are performing same function, like particular group of, see, here 5 cells are there. These group of cells are performing one function. Okay, so some function, one same function is performing by a group of cells. That means they are arranged into what? Tissues. So group of cells which are performing the same function, they are called as tissues. So they are having this coelenterate, they are showing tissue level of organization. Got it? First one is cellular level in sponges, but in coelentrates you can see tissue level of organization. So when we are moving to higher organisms like coelentrates and all, coelentrates and ctenophores and all, there you can see arrangement of cells that is becoming little more complex. From the basic level, it is moving to little higher level. That means the cellular organization is bit complex. So the cells, they have the same function and they are arranged into tissues. They are arranged as tissues. So it is called as tissue level of organization. In which one? In coelentrates, you can see tissue level of organization. At the same time, you can see cellular level of organization in sponges. But in coelentrates and ctenophores, you can see tissue level of organization. Next comes organ. Organ level of organization. This is organ level of organization. In higher phyla like Platyhelminthes and all, you can see higher level of cellular organization. Here, little more higher level of cellular organization. See, what is the change or what is the difference? Here, organ level is present. Organ level is present. That means tissues which are having similar functions, they are grouped together into organs. There we said in cellular level, some cells are performing particular function. When it is coming to tissue level, group of cells are performing a particular function. Right? When we are moving to organ level of organization, it is little more advanced. That means a group of tissues that is working as an organ that is performing a particular function. This is the tissue group. This is also a tissue group. So all these groups are performing a particular function. Got it? So the organ level is present. The tissues which are having similar function, they are grouped together into organs. So in animals, like animals of phylum, which phylum? Phylum Annelida, then Arthropoda, Mollusca, Echinoderms, and Chordata. We are learning about all these things in detail. That time it will become more clear. So they are in there. You can see in these kind of mollusca, chordata, arthropoda and all, you can see organ level of organization. So these systems, they are concerned with some kind of or specific physiological function. A particular physiological function will be performed by a specific organ. So that pattern, that is called as organ system of organization. We can say that digestive system in platyhelminthes, they have only a single outward opening of the body, and in platyhelminthes you can see a single outward of body. So this single outward that will work or that will function both as mouth and anus. Okay, so it is called as incomplete. That we'll learn. That topic is also coming. That point is also there. I'll explain. So in platyhelminthes and all, you can see organ level of organization. Got it? The animals like annelids, arthropoda, mollusca, echinoderms, and chordates. These organs have associated to form functional systems, and each system concerned with a specific physiological function. So that pattern is called as organ system level of organization. There is a particular organ to perform a physiological function. Okay. So that's the first basis of classification, which one? Level of organization. Next, feature. See here, organ system of organization. I'll explain one more point. We can see different groups of animals. They are exhibiting various pattern of complexities. See, we said about platyhelminthes, which are showing organ system level of organization. So in these digestive system, we can see a single opening to the outside of the body, and this opening, here you can see, this is working as mouth and anus. Okay. See, if one organism is there, just imagine this is the organism. Here only one opening is there. Only through this everything is going inside and it is coming outward. Okay. So there is only one opening for that whole body. It is working both as mouth and anus. So such kind of system, they are called incomplete. Okay, it is incomplete. Because for a complete system or a complete digestive system, you can see two separate openings. One will work as mouth and another one will work as anus. So it will go, food will go inside mouth and the waste excretory products will come through anus, right? So that is a complete digestive system. If one common body opening, outward opening, that is working as both mouth and anus, that is called as incomplete. A complete system will be having two separate openings as mouth and anus. Got it? Then circulatory system, same like that. Circulatory system also we can see there are two types of circulatory system, open type and closed type. In open type of circulatory system, blood is pumped out of the heart and the cells and tissues are directly bathed in that. Okay. In open circulatory system, you can see blood, it is pumped out of the heart. From heart, it is getting pumped and that heart is pumping blood, and the cells and tissues that will be, see, in the cells and tissues, this will be moving around. Okay, there is no proper channel around that cells and organs, cells and tissues. This blood will be moving. At the same time, in closed type, blood is circulated through a series of vessels of varying diameters, like arteries, veins, capillaries, and you can see here. This is closed. From here, you can see from heart, it is opening and it is moving around tissues and cells. Okay, but in closed circulatory system, what you can see, from heart, blood is getting pumped, and that blood is moving through small branches or small vessels, blood vessels, and in each organ, it will be moving. And again, that blood will be coming back to heart. Okay, so here you can see this kind of vessels through which blood is moving. Okay, that is called as closed type. Next, basis of classification. So based on level of organization, one type of classification, that feature we are considering for classification. Next is body plan. Based on body plan. See, you can see animals will be having different organs. So many organs will be there in animal body. Like in these body organs, these organs will be functioning together. But these organs, they are having different size, shape and all. So, based on that, there are three basic planes. What and all? Cell, aggregate, like body plans, cell aggregate, blind sac, tube within tube. Got it? So which and all? Three different body plans. So there, the organs of animals that will be having different size and shape. So all animals, they'll be having different types of body, and these are falling into three basic plans, which are the cell aggregate, blind sac, then tube within tube. Okay. So in cell aggregate, you can see cluster of cells. Okay. Cell clusters you can see, and these will be having labor division. This cluster of cells will be having labor division among them. And these cells, they'll be having no coordination, or sometimes they'll be having less coordination among them. So there is no tissues or organs in cell aggregate. There is aggregation of cells, just an aggregation of cells, but there is no coordination among those cells, or they'll be having slight coordination. That's all. But there are no tissues or there are no organs in that. So this type of body plans you can see in sponges. For example, cell aggregate body plan you can see in sponges. Okay. Next type is blind sac. Blind sac body plan. You have to learn about each and every term, because later while explaining about the features of each phylum or each group of organism, we'll be using these terms. I'll be telling about this will be having blind sac body plan or cell aggregate or tube within tube plans. That time you should be able to understand what is that. Next, second type is blind sac. Blind sac body plan, that is in blind sac body plan, that body, it is having only one opening in the alimentary canal. That is working as both mouth and anus. It is working both as mouth and anus. And there is tissue or organ system organization in the animals. In platyhelminthes, in coelentrates and platyhelminths, you can see blind sac body plan. Got it? Next type, third type is tube within tube. So what is that tube within tube? Tube within tube. That is those types will be having digestive system. Their digestive system as a continuous tube. And this tube that is opening at both ends. That is present in Annelida, Arthropoda, Mollusca, Echinodermata, and Chordata. You can see in all these phylums it will be present. Which one? Tube within tube. That means in that body you can see, just consider body as a tube. Inside that you can see another tube as a digestive system. Just for understanding, I'm saying like this. One tube, the whole body is one tube, and in that, imagine like that. Inside that you can see another tube like our digestive system. Okay, so digestive system is working as that is another tube, and that will be having separate opening at both ends. One is mouth and another one is anus. At the other end you can see anus. Okay, for example, nemathelminths and Annelida, Arthropoda, Mollusca, Echinodermata. In all these you can see this kind of body plan. Got it? Next basic feature of classification is symmetry. So in symmetry, based on their body symmetry, animals will be classified. So based on body symmetry, animals can be classified. Based on body symmetry, how we can classify? We can classify into three types. First one is radial symmetry, second one is bilateral symmetry, and third one is asymmetry. See, this is radial symmetry, radial. I'll tell you what is that. Then this is bilateral, and this is asymmetry. Asymmetry. So in radial symmetry, this one, the center one, this is the radial symmetry. So in radial symmetry, organism's body is divided in any plane. See, this is the central axis. Okay, the central axis. So when we are cutting or when we are dividing the body of this organism, and the division is passing through a central axis, when we are cutting like that, we will get, when it is getting divided through the central axis, you will get two identical halves. See, this is the central axis. When we are cutting like this, you will get two identical halves, right? This half also will be similar, this half also will be similar. If you are cutting in this plane also, both halves will be similar. If you are cutting in this axis also, this plane also, you will get two similar halves. So when an organism's body is divided in any plane and that division is passing across the central axis, means that body, in the dividing body you will get two identical halves. That is called as radial symmetry. When a plane passing through the central axis of the body divides that organism into two identical halves, it is called as radial symmetry. Okay. So for example, cylindrids, ctenophores and echinoderms. This is having radial symmetry. So note down the examples. This kind of questions only you will get. Radial symmetry. Among which one is showing radial symmetry? They'll be giving different phylums. So among that, cylindrids, ctenophores, echinoderms, these are showing radial symmetry. Next comes bilateral symmetry. What is bilateral symmetry? When that organism's body can be divided into a left and right halves which are identical only through one plane. Okay, only through one plane. That is the important point. That is called as bilateral symmetry. See here when we are cutting through this plane, the center, this plane. Here you can see the centered plane. You will get two identical halves. One A and B. Both are identical. But think about this organism. A coral polyp. When you are dividing only through the central axis, you are getting two identical halves. Right? Otherwise, you will never get. See, if you are cutting here, you won't get this half and this half as similar one. When you are cutting here, that also won't make two similar halves. Right? Only when it is that cutting or division is going through the central axis, then only you will get two equal or two identical halves. Here in bilateral, one beetle that is having bilateral symmetry. How? Only through one plane. If you are cutting through that central axis, that also only in one plane. That central axis, that division is going through central axis in one plane, you will get two similar halves. Okay. So bilateral symmetry, for example, you can see platyhelminths, then annelids, arthropoda, all these are showing annelids, arthropoda and all showing bilateral symmetry. Next is asymmetry. See, here you can see sponges. In sponge, there is no symmetry. Whichever plane you are cutting, either you are cutting this plane or this plane or this plane, whatever maybe, you will never get a two equivalent, two similar halves. You will never get. If you are cutting through any plane, whatever it may be, whichever plane it may be, you are not going to get two equivalent halves. For example, which one you can say? Sponges. Sponges are showing asymmetry. So for symmetry, based on body symmetry, we can categorize into three: radial symmetry, bilateral symmetry and asymmetry. Radial symmetry means through central axis, if it is passing, you will get two equal halves. Bilateral symmetry, only through one plane. If it is passing through the central axis, also only through one single plane, you will get two equal halves. In asymmetry, you are not going to get two equal halves. So for asymmetry, example sponges. For radial symmetry, you can say example cylindrids, ctenophores and all. Then bilateral symmetry, annelida, arthropoda. Okay. And next comes body cavity or coelom. Body cavity or coelom. First of all, what is this? Body cavity or coelom? It is the cavity or the space between what? Body wall and gut wall. Okay. The cavity or the space between body wall and gut wall. See, here, cavity is there. This is the space between body wall and gut wall. See, in this, here you can see this wall and this wall. You can see a cavity in between that cavity or coelom. Okay. So in a coelomate, among this, which one is a coelomate? Third one. This is the acoelomate. In this, they don't have animals which do not have the coelom. They are called as acoelomate. Acoelomate means they don't have the coelom or body cavity. They are called as acoelomate. For example, you can see coelenterate, flatworms, ctenophores. All these are acoelomate. Okay. So acoelomate means they don't have a coelom or a body cavity. So animals which do not have coelom. Next, second type is pseudocoelomate. These are pseudocoelomate. This type. Here you can see body wall and gut wall. Here also, see here also one cavity is there. This is like a cavity, but these spaces are filled. So this part is not completely empty. Here in between cells are there. Okay. It is not an empty space like this. So it looks like there is a cavity, but actually there is no cavity. This is pseudo. Pseudo means false. Coelom means cavity. So false cavity. Actually, there is no cavity. There is a false appearance of cavity. So the body cavity that is having incomplete lining of mesoderm. See this mesoderm that is incomplete. Here mesoderm, it is present but it is incomplete. So body cavity has incomplete lining of mesoderm. So instead of that mesoderm, what is present? Pouch-like appearance. Here you can see scattered pouch-like appearance is present. So in between ectoderm and endoderm. Ectoderm outside one, endoderm in between, endoderm and inside one. So in between ectoderm and endoderm, scattered pouches are present. These pouches are present. Okay. Mesoderm is absent or it is not completely absent. It is incomplete. Okay. So this type of body cavity you can see in this is called a pseudocoelom. For example, in roundworm you can see. Example, roundworm. In roundworm you can see pseudocoelomate body cavity. Next is eucoelomate. What is eucoelomate? Eucoelomate means the true coelom is present. true coelom. Here you can see these are coelomate. That means true coelom or true body cavity is present. This true coelom in that body cavity that arises as cavity in embryonic mesoderm. Embryonic mesoderm of that embryo, and that provides a cellular lining. Here you can see. Here this part is empty, but here you can see that cellular lining. So this true coelom, it is found in echinoderms, chordates, annelids and all. Echinoderms, in annelida you can see, and one more example we can see, chordata. In that also you can see true coelom. Got it? So this is the fourth feature which we are considering for classification. Classification of animal kingdom. Next is germ layers. Germ layers. What is germ layer? Germ layer means they are the first formed layers in the body. Germ layer means first formed layer in the body. And this layer is giving rise to all tissues, organs and all for the individuals. So based on this number of germ layers, animals can be classified into diploblastic and triploblastic. Diploblastic and triploblastic. So when the body cells, here you can see this is diploblastic and this is triploblastic. So body cells are arranged in two layers. There is an outer ectoderm, this one, outer ectoderm and inner endoderm. Here blue color that is mentioned as ectoderm and here inner endoderm, that's green color, green part, that is the endoderm. So like that two body layers are there. That is called as diploblastic, or that type of animals, they are diploblastic. Got it? Here, at the same time, you can see here endoderm, inner layer, ectoderm, the outer layer, and in between these two, you can see a mesoderm. That means three germ layers you can see. Here in triploblastic, you can see three germ layers. Here, two germ layers. Okay. Ectoderm, endoderm, and in between ectoderm and endoderm, you can see mesoderm. Okay. So in diploblastic, ectoderm and endoderm is present. In triploblastic, ectoderm, endoderm, and in between these two, you can see a mesoderm. Three germ layers are present. Which are they? Ectoderm, endoderm, and mesoderm. Here in diploblastic, you can see ectoderm and endoderm. Okay, two germ layers are present in diploblastic. So the germ layers, that is one feature which we are considering, or one character which we are considering for the classification of animal. Okay. So there are some more characters which we are, or some more features which we are considering, like respiratory system and all. So that we'll learn in the next class. So around 12, 13 characters we are considering for classification of animals. So up to this, hope it's clear. Just go through this, because you have to make sure that you're clear with this, because later while explaining about each phylum, we'll be using these kind of words. The animal, that this group of animals, that they are diploblastic or they are triploblastic, like that only we'll say. So that time you should not get confused that what is triploblastic or what is diploblastic or what is called a germ layer. So it should be clear. Okay, got it? So that's all about today's class. If any doubt you have, you can ask me in the next class. Thank you.